Hello children, in this part we are going to talk about the gram staining and how by using this gram staining we can differentiate between gram positive and gram negative bacteria. So here first we will differentiate between we'll see the differences between the gram positive and gram negative bacteria, and then after that we will see the staining procedure. So now if you see that if we are going to stain bacteria and this bacteria takes the stain, so then that will be gram positive bacteria, and if it is not going to take the stain, then it is known as gram negative bacteria. So before we enter into the process of gram staining, we will first see the structure of gram positive and gram negative bacteria. Now if we see, in case of gram positive bacteria, we see there is this is a membrane, that is the plasma membrane, and outside this is a cell wall present. So here in case of gram positive, the cell wall is little thicker because there are this membrane. This consists of peptidoglycan and there are number of membranes in it, so that is thicker in case of gram positive. And this cell wall, it also contains peptidoglycan in it, which consists of teichoic acid, and it is very important. Whereas if we go for gram negative bacteria, what we see here, that the cell wall, as in comparison to the gram positive bacterian cell wall, it is thinner. And here also see, first is the plasma membrane, then that is covered by cell wall. This cell wall is thinner. This also contains peptidoglycan, but here teichoic acid is absent. So what we can see from here is that there is a difference in gram positive and negative bacteria on the basis of presence or absence of teichoic acid in the cell wall. Okay, and here if you see the membranes of this peptidoglycan, that is thicker as comparison or as compared to gram negative bacteria. So that is why one reason that cell wall is thicker in case of gram positive, and it contains teichoic acid in it, whereas there is no teichoic acid which is present in case of gram negative bacteria. There is an additional membrane which you can see here, which surrounds the cell wall, and this additional membrane, this is made up of lipoprotein. So there is one more you can say phospholipid layer which surrounds this gram negative bacteria. Now it is clear that gram positive and gram negative bacteria, they differ in their composition of cell wall and in their functioning also. And why this is so? Because gram negative has lipoprotein, phospholipid, and it also has polysaccharide. So all these things, since they are present, and that is one reason why there is a difference in their staining also. So the one that takes the stain and the other one does not take the stain. So the one which takes the stain is gram positive, and the one which does not take, that is gram negative. Now let us see the procedure of gram staining. So what we do here is that first of all we heat this bacteria and we fix it on a slide. So the first step is heat fix bacteria on a slide, and then the second step is that we pour crystal violet to it, and that is a kind of stain, you can say, which helps in coloring. So then we leave it for say about 30 seconds or up to a minute, and after that we have to wash it. Now once we wash it, then we have to add this iodine, and this iodine is the gram iodine. It is called as gram iodine. We add this on the slide, and again we have to leave it for 1 minute, and then after that we wash. So before we wash, what we see here, we have to wash it with acetone or alcohol. But before that, what we see, that all bacteria, they turn purple. So we will wash it. We will take this 90% of alcohol or this acetone, which helps in the washing. is observed is that all these bacteria, they turn purple here, and after washing it with the acetone or alcohol, what next we observe is that some bacteria, this purple stain, it retains, and the ones in which it retains, that is called as gram positive bacteria. And because the cell wall which is present in it, it takes the color and it helps in retaining of the color, whereas at the same time we see that some bacteria, they lost the color. So before washing it was there, but once it is washed with acetone or alcohol, they lose the color. And then we add saffronin, that is another kind of stain, to it, and what happens is that this will also give color, but then gram negative, they take this color which comes through from saffronin, and they do not take the color that was added by crystal violet. So since there is a lipoprotein layer which is present, so due to lipoprotein layer, no color is taken by this iodine purple, and afterwards when the color is lost, so they take this saffronin color. So this is the whole procedure that talks about the gram staining, and it helps in differentiating between gram positive and gram negative bacteria. So I would like to tell you one more thing over here, that this color, why it retains in gram positive, because there is a cell wall that is made up of peptidoglycan, and in case of gram positive, there is no other layer that has this this peptidoglycan layer is not covered by any other layer, whereas in case of gram negative, this peptidoglycan layer has a cover of lipoprotein. So if you see this peptidoglycan, now when this color reaches down here, it also takes the stain, but then what happens when it is washed, then this lipoprotein layer, since it does not retain the color, so it washes off, and then on this basis that this peptidoglycan, it retains the color in gram positive, so the name gram positive is given. So this helps in clear distinction between gram positive and gram negative bacteria. See, this gram negative bacteria, though they take the stain, but they take the saffronin stain, and whatever color was present here was taken up only temporarily by this layer, that is lipoprotein, and it gets washed off easily by acetone or alcohol. And when it is further treated with saffronin, so it takes that red color. So see, if the cell wall which has peptidoglycan, it has only peptidoglycan, then only this crystal violet will be present there in that, and it will give the purple color. And when this peptidoglycan also has one more layer over it which covers it, that means it has a this lipoprotein layer which is covering it. So what will happen, that it will not take up that stain. In place of that, it will take some other stain, that is saffronin. Also, the important thing is that gram positive has teichoic acid, and gram negative, they do not have teichoic acid. Now these gram positive, they are more harmful. Why is it so? See, all these chemicals and drugs, they are strong, and we need to control the bacteria by giving this strong chemicals. So these stronger chemicals are required to kill this bacteria. So it takes time in them to reach or act inside the actual structure of bacteria, because they consist of lipoprotein, and they are mainly disease causing organisms, example Vibrio. So this lipoprotein layer, it actually protects bacteria from all these chemicals if it is subjected to. So now if we see, there is a difference between this cell gram positive and gram negative bacteria. So if we see the cell wall, that is very rigid in case of gram positive, whereas elastic in case of gram negative. Then penicillin susceptibility, it is very high in gram positive and it is very low in gram negative. Then nutritional requirement, it is complex in case of gram positive, and it is simple in case of gram negative. And the last difference is that physical disruption, so it is more resistant for the disruption, gram positive, and it is less resistant for gram negative bacteria. So this was all about the difference between gram positive and gram negative, and the staining, and depending upon the stain, that whether it takes the stain or it does not take the stain, we are able to differentiate between gram positive and gram negative bacteria. And this gram staining was discovered by Hans Christian Gram. So I hope this part is clear. Thank you. We will continue with the next in the next video. We are going to talk about respiration. Thank you, children.